Okay, thank you, and it's always a pleasure to be here. This is one of the highlights of my year. So I think this talk might better be titled, but first off, the guy with hair there is me a few years ago in my pre-Colorado look. That's Greg Wilson, who was a big part of this as well, and he sends his regrets to everybody that he couldn't be here. And this is Gunther Schneider, for those of you that don't know him. But we thought we would better title this Tale of the Three Little Pigs. So this is a study that we did several years ago, and I'll give you a little bit of background about a lot of what I've talked about at this society for a number of years now, which is really looking at what happens to the relaxivities of gadolinium contrast in human blood. And it's very different in blood than it is in plasma, and it's also very different at the very high concentrations that we achieve during first pass. And if we understand this, as we think we do now, then you can really model, predict, and optimize contrast-enhanced MRA. But part of the whole goal of this study was to try to validate this in vivo. So just by way of review, if we look at the relaxivity, the R1 versus concentration for pretty much any contrast agent, as we get to these very high concentrations, like we see in blood during first pass, you get a deviation from linearity. And this has to do, we've shown, with the water exchange across the red blood cell membrane itself. So an intrinsic property of blood that basically robs you of as much relaxivity as you might think is there. And then T2 star, or R2 star, is a very important thing as well. And again, this is very different in blood than it is in plasma. And a couple of things to just note. So I've got 1.5 T on the left, 3 T on the right, blood on the top. And one thing you notice is that the R2 star, that the slope there is much greater for blood than it is for plasma. It's also much greater at 1.5 T than it is at 3 T, and but interestingly, for all the agents, it's relatively similar, which all comes out of the physics, and we've published this as well. And these effects are very relevant for first pass contrast MRA because, for instance, if we hit a 10 millimolar gadolinium concentration, which we do very easily, the T2 star is on the order of 2 milliseconds. So that's going to take away a lot of signal. And if you understand these things, then you can do modeling that looks like this, where there are 4 types of contrast that you can see we put up there in different colors. And you can look at how much signal intensity you would expect versus injection rate. And we have what we call the front side of the curve, where the faster we inject, the more signal we get. But then you hit a plateau phase, and ultimately you hit a backside where actually injecting faster is not good. And just instantly look at where 2 cc's per second falls here. You're really on the backside of the curve for a lot of contrast agents. So one of the things I've talked about over the years and tried to get people to understand and to do with their MRA is that really at physiologic cardiac outputs and the doses we give, injecting anything much faster than about 1 ml per second isn't really going to help you. It's probably going to decrease your signal intensity, and what it ends up doing is it ends up effectively shortening your bolus because you've injected the contrast faster than you need to. This is just an example from some clinical cases where I talked our neuroradiologists into dropping the effective rate from 2 mLs per second to a half an ml per second. You'll notice there's not much signal intensity difference, and the image on the left is certainly quite a bit blurrier due to that shortness of that bolus at the faster injection rate. So the whole goal of what we tried to do that I'm going to explain today is to validate this type of modeling in an animal system to really confirm that the work we've done really works in vivo to accurately predict signal intensity, and also to kind of show that this signal plateau that I'm talking about at about 1 ml per second is very real in an animal system. And thereby kind of validating that the slower or diluted, if you will, contrast injection is a nice way to go. So the study was, this is done in conjunction with Gunther Schneider in Hamburg, Germany. We took 8 juvenile pigs, 50 to 63 kilograms on a Siemens era. These pigs were all anesthetized. We did multiple MR acquisitions during gadolinium injection. One of the first things we did is just harvest some of their blood, repeat the studies I showed you in the beginning on human blood, looking at T1 and T2 star for these 4 different agents by looking at look-locker and multi-echo FFE sequences, seeing basically the same thing, that there's deviation from linearity of the relaxivity curve of the R1, just like we saw in human blood. And R2 stars, again, is at 1.5 T, but they were very similar for all the different agents and significantly greater in blood than they are in plasma. Again, this has to do with not only water exchange, but just the heterogeneity of blood itself. We then took blood at different concentrations and ran MRA type SPGR sequences and looked at the signal intensity we got versus contrast for concentration for all 4 contrast agents, as you can see there, and got pretty nice fits from our modeling to what the reality was, but we're still ex vivo here, right? And we did see the linear, the plateau, and the backside of the curve as the contrast concentration increased, as is shown. Now, for these animals, what we did is we took 4 different contrast agents, 8 animals, and we injected 2 of them with each gadolinium agent in different sessions. And in each animal, we did a time-resolved SPGR MRA type sequence with the parameters as shown, and that ended up looking like this, 1.74 second resolution, so we can get time versus signal intensity curves like this. And then we injected them with 1, 2, and 3 ml per second injections of full dose each time, about 15 minutes in between boluses. The order of that injection rate was randomized, and 3 boluses per animal. And the interesting thing about this study is what we simultaneously did is we had an indwelling aortic catheter, which we were able to sample the arterial blood at 2-second intervals for the purpose of doing gadolinium mass spectrometry so that we can know what the gadolinium concentration is along with the signal intensity. So here's just an example of Gunther Schneider's team with the pigs in the suite. Here's a couple of examples just looking at 1, 2, and 3 ml per second injections with, in this case, multi-hance, and they were done in different orders. These are on the 2 different pigs, but the thing to notice here is that the faster injections, the black and the red, aren't giving you any more signal intensity, they're just giving you shorter acquisitions or shorter plateau periods. Here's one with gadoteridol. Again, similar thing, the faster injections aren't really buying you much more signal intensity, the slower injections buying you a longer plateau, and that can be seen in this graph where the red line is the relative signal intensity as we go from 1 to 2 to 3 mLs per second, and the blue line shows you that we're actually decreasing, all we're effectively doing is decreasing the length of that injection, which then leads right to blurring. We did we did 2 additional contrast MRAs on each pig, where we injected either at 1 cc or 3 cc's per second, full doses again, in a random order, and did an actual MRA. And many of you in this audience, I sent a few of these pictures to. So we have kind of 8 pairs here. I'm not going to show them all, but I basically asked a lot of you, and thank you for those of you who contributed. This is a kind of a non-scientific way to do this, but which is sharper, A, B, or the same? And one of these is 1 ml per second, the other's 3 mLs per second. So I can kind of ask you to look at them yourself here, and I'll show you the results of the 12 experts in a moment. But which is sharper? This is blurring effect, right? And you also notice there's really no signal intensity difference between the 2 of them. And these are the crossover results. So the blue lines are the number of times that the readers said that the 1 ml per second was better. The red is the time the 3 ml per second, and green were where people really couldn't make up their mind. And particularly for the last 5 animals, you can see there's a pretty overwhelming preference for the 1 ml per second. study. This is based on blurriness. Now, as I said, this is kind of an informal, we'll get to further analysis later on, but this is pretty statistically significant here. And of course, the biggest part of this is the mass spectroscopy. And I just have to apologize because I'm told those are the Swiss Alps. And they lie between Germany, where all this data is, and Italy, where the mass spectroscopy is to be done. And this data has been deep frozen in Gunther Schneider's freezer in his lab for I don't want to tell you how many years yet. But we're finally, I think, wading through the red tape and the bureaucracy and getting that transferred down to a lab in Milan where it can actually be analyzed, and then we can kind of validate all this. So next year, hopefully, I'll give you, now that we've got this introduction, I'll let you know kind of what that ended up being. So the point here is that it's important to understand gadolinium-based contrast agents are very different in blood than they are in plasma or in any other kind of fluid, particularly at high concentrations, and that R2 star can be very, very high, particularly at 3T, and that leads to this signal plateau or loss. And really, if you put that about 1 ml per second number in your brain, I think that is a pretty good one, because going beyond that is usually not going to help. So we're awaiting that mass spec data so we can correlate time-resolved signal intensity with the gadolinium and make sure that we have a good in vivo validation of the theory that we've been talking about for a number of years. So with that, I'll show you a picture of my new toy, which I'm happy to talk to anybody about at any time. Thank you.